Welcome everybody to another Frontiers in Brain Health. We're excited to once again have a very relevant topic that combines the mind and brain. I'm Dan Krozik, the Deputy Director here at the Center for Brain Health, and welcome to all of you, as well as welcome to our online audience. We have people out there who are going to watch this talk virtually. And if you're doing so and a question arises, go ahead and drop that in the Q&A function, and we will get to some of those online questions as well as some of your questions here. And today we have Dr. Andrew Eagle. He is an expert in motivated behavior and how it's carried out within the brain. He did most of his training at Central Michigan University from his bachelor's all the way up to his PhD. He moved to Wayne State Medical School where he did a postdoctoral fellowship and then moved to Michigan State. And he was a research assistant professor at Michigan State. Some of his work that he'll talk about today was actually from up north there. And he joined UT Dallas in 2024 in our Department of Neuroscience, where he is an assistant professor. And his work is very detailed, examining the role of numerous brain regions in processes such as memory and guiding our behavior, which is all very relevant and very exciting. He uses a diversity of cutting-edge methods to address his research questions as well, and those are relevant to many health conditions, including exposure to post-traumatic stress, chronic stress, aspects of substance abuse, and probably relevant to much of our mental health. And we're very excited to have him here at UT Dallas. He's a prolific mentor, and I was chatting with him about this before the talk. He shares my belief that it's all about training those future scholars and building our brain capital in that way. He's also a really interesting teacher who's taught a variety of different courses, all related to brain and mind. I'd like to take some of those courses myself. And without further ado, let's go ahead and welcome Dr. Eagle.
Thank you very much. Do I need to be on mic, or can everyone hear me?
For the recording.
I can project myself pretty well.
For the recording.
Hello? Okay, great. All right, well, I won't need to keep this too close to me, so. All right, just want to say thank you for everyone for having me in this wonderfully beautiful building, in this room. As I was saying earlier, I've come here before for various grad program things, and it's always such a nice building, so it's always nice to come here. And I just thank you all for coming here to my talk. So let's talk about my talk. As Dan said, and thank you for that introduction, Dan. So I'm interested in, this is going to be more like a memory talk. And I'm really interested in how memories shape and guide future behavior, and how things like addiction and depression, mostly I'll be talking about addiction today, but addiction and depression and things like PTSD, how they can hijack those same memory systems so that you have very, very strong memories for drugs or strong memories for like stressful events, and how those can inform or contribute to various neuropsychiatric diseases. Okay. And particularly, I'm going to be focusing on this one particular region, this entorhinal cortex. So this is one major project that we're doing in our lab, and I'm really excited to talk about it. But before we start talking about drug memories, let's first define memory. So declarative memory includes those memories that can be brought to consciousness and expressed as remembered events, images, sounds, and so on. And these types of memories can be expressed through language, aka declared, and you can also measure this in non-human animals. So in those animals, declarative memory typically refers to information that the animal is aware of and could be declared if they had the ability to talk like we do. And declarative memory itself is dependent on the integrity of the medial temporal lobe, as you can Is this not working? Oh, it doesn't very help much. Okay. All right, well, you can see the medial temporal lobe here. Oh, yeah. It's good. But you can see the medial temporal lobe here. The system consists of the hippocampus, or the hippocampal region, and the adjacent perirhinal, entorhinal, and parahippocampal cortices. And I won't talk too much about the non-declarative memory. So memory is also temporally segregated across time. And this includes sensory, aka immediate memory, short-term memory, and long-term memory. And a major driver of consolidation from immediate memory becoming short-term and short-term becoming long-term is the major driver of that consolidation is the progressive stabilization of memories that follow the initial encoding of these memory traces. And all of that relies on things like changes in gene expression and protein synthesis. And if you block gene expression during these key periods of time when the memory is being encoded, you can block that memory. So I want to point out how important sensory or immediate memory is. So a key feature driving memories to be consolidated is their meaningfulness or their salience, so how important they are. So important events might be more likely to be consolidated compared to less significant events. Whoops, just hopped ahead. And memory is incredibly useful for our survival, being able to note and remember our sources of food, avoidance of predators, etc. However, memory, as we see, can go horribly wrong. And we see that in neuropsychiatric diseases and neurological disorders like Alzheimer's, post-traumatic stress disorder, depression, addiction is where we see impairments in memory. And our lab is particularly interested in how these memories go wrong in addiction. So what is addiction? So addiction can be defined as a chronic disease characterized by drug seeking and use that is compulsive or difficult to control. And they do this despite these harmful negative consequences. And it can have massive impacts on society, including crime, lost productivity, increased healthcare costs, and death. And so this graph is from the CDC, CDC Wonder. And you can see here, since 1999, we've been tracking the number of overdose deaths across a range of drugs. And you can see here that cocaine overdose deaths are on the rise, as well as other psychostimulants. They're in orange and yellow. Now, you may or may not be working on the assumption that cocaine use is down. I have heard people tell me this. They say, I thought cocaine was no longer a problem. And that was true in the early 2000s, after the crack and cocaine epidemic, where we saw a lot of cocaine use go down. However, as you can see here, overdose deaths are increasing, and there are additional data showing that cocaine use is increasing. The stimulants are back on the rise again. And these numbers aren't just data on a graph. These are real people that are dying. And while we do see some overdose deaths from cocaine, as you saw in that last graph, many of these are likely related to polysubstance use, like using more than just cocaine, or cocaine laced with fentanyl. However, people with cocaine addiction or cocaine use disorder, as it's clinically diagnosed, they rarely die from overdose. But they're still dying. They're just dying in their 30s and 40s and 50s because their vasculature is terrible, which puts a massive burden on healthcare costs to the individual and society. And also over the last decade, we're observing increases in prescription stimulant abuse from drugs like Adderall and Ritalin, which are very common in the college Gen Z population. But we're also observing this in older adults and especially among women. I've got more bad news, though. Sorry. Recent news reports from February of this year, so just a few weeks ago, indicate that we are looking at a potential cocaine boom due to a massive increase in the global supply of cocaine, which will inevitably lower the price of cocaine, and that may increase its use here in the US. And so I predict that we're primed for a major uptick in cocaine abuse. And then lastly, another bit of bad news. There are no FDA-approved medications for cocaine use disorder. And so together, rising availability, abuse, and mortality, and the absence of any pharmacotherapy, any drugs, underscore the urgent need to define neurobiological mechanisms that could guide that therapeutic development for cocaine use disorder. So let's talk about that neurobiology that underlies addiction. So one of the main individual brain regions in this addiction is the nucleus accumbens. So the nucleus accumbens is a deep midbrain region that is incredibly important in motivated behavior, or the drive to seek out rewards such as food, water, sex, social interaction, and the drive to avoid noxious stimuli that can be painful or lead to death. The nucleus accumbens can also become dysregulated by drugs of abuse due to leading to plastic changes that underlie addiction. It also acts as a hub of information from a variety of cortical and subcortical regions that are all kind of centering on and projecting their information to the nucleus accumbens. And these include regions like the prefrontal cortex, the medial temporal lobe, including the hippocampus and entorhinal cortex, and the amygdala. And these regions convey different aspects of information that to the nucleus accumbens that drive motivated behavior, and these neurons that are projecting from these other brain regions. We're talking about brain cells, neurons that are sending their projections to the nucleus accumbens. These neuronal projections can become similarly dysregulated by drugs of abuse and lead to addictive behavior. So let's talk about what we know about addiction. Addiction can be defined across 3 stages in a cycle. The binge intoxication stage, which is defined as uncontrolled use. The withdrawal stage, which is defined as negative affect and other physiological symptoms from early abstinence, like typically just a day or a few days after abstaining from a drug. And craving, which is defined as a preoccupation of thoughts on their chosen drug and anticipation or craving to seek out and procure drug. Now, as you can see here, this is a really complex figure, I understand. And what you can imagine is that means that addiction itself is a very complex disease, which is why it's so hard to treat. And it's regulated by many different brain regions that mediate different aspects of these stages. Now, we won't discuss all of these, but I'd like to focus on that craving part. So one of the major predictors of relapse to drug taking and drug seeking is the amount of craving. So if we can target and reduce craving, we may be able to lessen the probability of use. And we know that things like cues and contexts drive craving, both in natural rewards. Everybody's eating right now, so hopefully nobody's deriving any craving from these images. And we also see the same thing for drug rewards. And people, places, and things can also act as various cues associated with drug taking. And so all of these cues are represented in these various memory regions and then can drive motivated behavior. So thus cues can drive craving, which leads to a higher probability of relapse, as you see in this unfortunate individual on the right here. And then this thereby increases that association of that drug reward with that cue, strengthening this drug cue associative memory, a memory where it's associating this cue with this drug. And then that may help facilitate future addictive behavior, like drug seeking, creating this vicious cycle. Now, I'll be focusing our talk on these memory-related regions, including the entorhinal cortex. Now, as I discussed before, the medial temporal lobe consists of the hippocampus and entorhinal cortex. These two regions are incredibly important in the consolidation of memories. And there's a lot of interconnections between these two regions, so that the hippocampus is talking to the entorhinal cortex is talking to the hippocampus. And this is incredibly important in that consolidation, meaning the transfer of those short-term memories to longer-term memories that are then essentially those memories are distributed across the rest of the cortex. And so in these regions, the hippocampus and entorhinal cortex can consolidate memories of things like stress and drugs with the cues and contexts in which they occur. And later, these stress and drug-associated cues and contexts can reactivate these memory regions and others, and then that can lead to drug craving. That activation can lead to drug craving, which is a major driver of drug relapse. So understanding how this process works and how we can disrupt this system is critical for treating addiction. And if we can target these drug memories, or these drug cue memories, or drug context memories, without affecting all of the rest of the normal memories, like what I had for breakfast, or what do I have to do tomorrow, all of these other things, just targeting these drug memories, then we may be able to reduce relapse. Now, the entorhinal cortex, see if I can do this with the mic in my hand. The entorhinal cortex can be functionally segregated into two regions, the medial and lateral regions. Now, the medial entorhinal cortex some of you may have heard of things like place cells or grid cells. These are cells that basically respond to a place in the environment. So the medial entorhinal cortex is really important for spatial mapping the environment. I'm not going to talk too much about that. The lateral entorhinal cortex, or LEC, processes non-spatial information, such as objects and sensory cues, and relays that information to the hippocampus to support memory and associative learning. Now, the LEC encodes information about things like navigation and time, objects and contexts, and it's incredibly important for encoding these many different aspects of the environment, like cues and objects, where the objects are in a particular context, their place. All these different aspects get associated together. And it's important, the LEC is important in the consolidation of these associations, of these different aspects of the environment, and we call this associative memory. So like the hippocampus, the LEC also contains a population of neurons that send axonal projections to the nucleus accumbens. So, neurons are like one of the primary brain cells. They look like a tree. They have this long axon. Some of your axons throughout your body can be up to like 3 feet long, 3 or 4 feet long, depending on how tall you are. In the brain, they don't have to go that far, but essentially these axons from the cell body can go to other different regions and synapse onto other neurons in that other region. So these LEC neurons contain what we call those neurons that go from one brain region to another, that send their axons from one brain region to another. We call them projection neurons. And the LEC sends projection neurons to the nucleus accumbens. And this has been known for almost 30 years. However, the function of these specific neurons in the LEC that send their axonal projections to the nucleus accumbens, the function of this pathway is completely unknown. However, we do know about projections from the hippocampus to the nucleus accumbens, the prefrontal to the nucleus accumbens, and the amygdala to the nucleus accumbens. We know we haven't closed the book on them. I don't want to say we know everything there is to know about those other projections, but we know quite a bit. But for this circuit, it's never been studied in terms of its function. And furthermore, early studies have indicated that when cues are paired with cocaine, those same cues can later activate the LEC. So some very early work from fMRI imaging studies showed that cocaine-associated cues, like pictures of crack pipes or cocaine powder like you see here, increased activity in the entorhinal cortex in cocaine-dependent subjects compared to neutral cues. It was also previously observed that cocaine-associated cues led to greater activation of the LEC in cocaine self-administering rodents. And more recent evidence has shown that the LEC responds to pleasurable stimuli like rewards, noxious stimuli, and cues associated with these stimuli. So these include, and I'm not talking about the projection neurons, I'm just talking about the LEC brain region in general. And so these include odor cues associated with foot shock, odor cues associated with natural rewards like sucrose or food, navigational cues associated with natural rewards, and another study showing odor cues associated with reward. Thus, there is strong evidence to suggest that the LEC may be conveying information about the salience, the importance of these various cues and contexts associated with positive and negative outcomes. However, that's just the LEC in general. Whether these subpopulation of LEC neurons that send their axonal projections to the nucleus accumbens similarly encode this information is unknown, as these previous studies did not solely target those projection neurons. So, with all of this in mind, we are interested in understanding the role of these LEC to accumbens projection neurons in motivated behavior. We know these projections exist. We know that the LEC is activated by drug cues, as well as cues associated with positive and negative stimuli. However, the function of this subpopulation, like there's lots of LEC neurons in this, but not all of them project to nucleus accumbens, only some of them do. In that subpopulation, the function is unknown. So specifically, whether these projection neurons regulate motivated behavior. So what we're asking is, how and when do cocaine cues and other natural reward cues activate these LEC neurons that project to nucleus accumbens? And we have two ways that we go about asking that question. So we use a molecular marker, CFOS, which I'll explain here in a bit, to detect neuronal activity. And then we also use this in vivo, in animal calcium imaging during food and cocaine self-administration, where essentially a rodent, in our case a mouse, is working to get food or cocaine. And we do that while we're doing calcium imaging, and I'll explain that as well. Then we also ask, so how do I tell if an engine is necessary for a car? I can take it out and it doesn't run. That tells me it's necessary. That tells me about its function. And so we ask those same functional questions. Are these LEC projection neurons necessary for relapse to cocaine seeking? And so we use these chemogenetic and optogenetic approaches to essentially acutely turn off this circuit during cocaine self-administration. And we also do it during other tasks as well, which we'll go over. All right, so I first set out to study this population while a research assistant professor at Michigan State University. So this early project was conducted entirely by me and some really excellent undergraduate researchers. So my students, Grace and Brianna, first conducted a study to determine whether these LEC projection neurons are activated by salient contexts. To achieve this, we measured CFOS, which is a molecular marker or a protein marker for recent neuronal activity. CFOS is an immediate early gene that in response to high neuronal activity, so usually it's at very, very low levels, very, very low levels. But in response to high activity in a neuron, it massively increases its expression within 30 to 90 minutes, and then is mostly gone by about 4 hours. So it's kind of a time window into when neurons are recently active. We can then take brains from mice that undergo various tasks and conduct histology assays in brain tissue slices to determine which cells were specifically active during a period of time, indicated by which cells are expressing this CFOS marker. We also sought to label or mark those LEC projection neurons. We can't just see them with the naked eye. And to do that, we relied on these genetic mouse lines that express green fluorescent protein, or GFP. It's a protein that fluoresces green. The way we did that is we have these L10 GFP mice that essentially have this inserted gene that expresses GFP. I'm going to use my pointer here for this. All right, so here's our mouse line. So they express this gene here. It's called this L10 GFP gene. So when we insert that gene into these mice, they're not expressing GFP in any of their cells. And the reason why is because it has this thing called a stop codon. The stop codon basically prevents the gene from being transcribed. It prevents RNA polymerase from reading that gene. So they're not expressing any GFP. However, this stop codon has these two little indicators on either side of it. They're called LOXP sites. Well, there's this enzyme, it's called Cre recombinase. What it will do is when it's expressed in a cell, and Cre recombinase is not normally expressed in any of your cells, so don't worry about it. Cre recombinase will pull out any genetic sequence that's between those two LOXP sites that you see up there. It will take that out. And what it will take out, in this case, is the stop codon. And then any cells that have that gene where the stop codon is removed will start expressing this green fluorescent protein. And what I can then do is target the expression of which cells I want to express that Cre recombinase that's going to remove the stop codon and allow this GFP gene to then be read by RNA polymerase. So basically, wherever there's Cre in these mice, there will be GFP. So then how do we get Cre into just our LEC projection neurons? Well, we don't really. What we did is we injected a retrograde recombinant viral vector. This is essentially a virus. It's not a virus that's going to make you sick. It doesn't replicate. Most viruses make other viruses. In this case, we've engineered them so that they only make a gene of interest that then the cell will take up and allow that gene to be expressed. So we used, and it's a retrograde, meaning that it gets taken up by the axons, and then it can move its way. So it gets taken up here by the axons, and then moves its way up into the soma, or the cell bodies of these LEC projection neurons. And then this virus expresses that Cre recombinase. And so the viral-driven Cre recombinase drives the removal of the stop codon in these projection neurons, and this now allows the gene to express that fluorescent protein GFP, and now we have GFP positive projection neurons. We can see them. And essentially, we've tagged these LEC projection neurons with this fluorescent reporter. So now we know, now we can see which ones are the LEC projection neurons and which are not. So my undergrads, Grace, and grad student, Brianna, then conducted a study to determine whether context paired with cocaine would activate these LEC projection neurons. So they compared these naive L10 mice that were left in their home cage. They also got the labeling viral technique so that they could, so we could label their projection neurons. Other than that, they were just left in their home cage. Now, these animals shouldn't have any CFOS expression because they haven't been exposed to anything salient. They're just sitting in their home cage all the time. We compared that to mice that were exposed to either cocaine or saline in 2 different contexts over 2 days. We'll call that cocaine CPP. And to mice that were exposed to saline in 2 different contexts. And we'll call that saline CPP. So basically, one group got cocaine half the time, another group never got any cocaine. And 1 hour after the last drug context pairing, or saline context pairing, the perfused brains were collected for CFOS immunohistochemistry. And this is what the immunohistochemistry is going to look like. I'm going to use my laser pointer here. All right. So what you're seeing here in purple is a window into the LEC showing all of the cell bodies in purple, and all of those cell bodies are the neurons that express GFP, which are only going to be the LEC projection neurons. So wherever you see purple, those are the LEC projection neurons. Here, these cyan cells, those are all cells that express CFOS. Those are highly active cells. And then this is just both of those 2 images combined. And all of the arrows indicate essentially the LEC projection neurons that are highly active. And they essentially went through and counted all these cells. It's a really fun thing to do. And what they essentially took is all those cell counts, and they looked at the number of total GFP positive cells. So there's no difference in the amount of projection neurons between the groups, which is exactly what we would expect. However, we saw an increase in the total number of CFOS positive cells in both the saline CPP group and the cocaine CPP group. And then this is just general LEC. This is not specific to the projection neurons. This is quantification of those overlapping, like the arrows, those LEC projection neurons that were recently highly active. And again, we see really no difference between the saline CPP and cocaine CPP mice. These results indicate that contexts, regardless of what occurs in that context, activate the LEC and these LEC projection neurons. This was surprising to us. We expected that the cocaine-paired context would lead to greater activation compared to the saline-paired context, but clearly it did not. Oops. In the right spot? Okay. All right, they next tested whether a context paired with negative stimuli, like a mild foot shock, would also activate these LEC projection neurons. So they placed mice in a novel context where mice were exposed to 3 mild 1-second 0.8 milliamp foot shocks over the course of 5 minutes, and then a separate group of mice were exposed to the context alone without any foot shock. And we found that similar to the cocaine context experiment, that contexts, regardless of the presence of foot shock or not, activated the LEC projection neurons. And so we see that here. We see that both the ones that got foot shock and the ones that just were exposed to the context alone, no foot shock, had increased CFOS expression as well as CFOS expression in our projection neurons. So this could indicate that the LEC activity is conveying signals about the salience of an experience, particularly a novel context, but the LEC, including the LEC projection neurons, may not be encoding other information about the stimuli, like its valence, whether it's pleasurable or aversive. And so we next wanted to know whether these LEC projection neurons are necessary for encoding these salient context memories. So to do this, we used a chemogenetic approach. These chemogenetics is using these DREADS receptors, or designer receptors exclusively activated by designer drugs. Yeah, it's a mouthful. Essentially what we're doing is we're taking these engineered receptors. These are G protein coupled receptors. And we can insert them into any cell that we want using either those viral techniques or genetic mouse strains. We can insert those receptors into any cell. And what those receptors do, they can either inhibit the activity of that cell that it's in, that neuron that it's in, or it can activate that neuron. That's what these dread receptors do. Now, normally receptors, like all the receptors that we have in our body, they usually have a ligand, some type of chemical that would bind to that receptor and cause it to be active. In this case, these don't have an endogenous ligand in the body of mice, right? So what we can later do is we can give a systemic administration of the ligand for these specific dread receptors, a designer drug that will only activate or exclusively activate these designer receptors. And so therefore, we can use chemogenetics. Essentially, we can turn off or inhibit a specific set of neurons, in our case, the LEC projection neurons, and we can use this chemogenetics tool to address our hypothesis that inhibiting these LEC projection neurons will impair cocaine and fear contextual memories. So let's see. So we use this approach to test the necessity of these projection neurons in cocaine context associative memories. So my undergrads, Haley and Luis, tested the necessity of these projection neurons in cocaine conditioned place preference, or CPP. Now CPP, that's this box you see right here, is an established assay to measure reward and memory and consists of a 3-chamber box where each chamber is contextually distinct, different floors, walls, etc. And over the course of 2 days, we confined mice to each chamber with either saline in one chamber and cocaine in the other chamber. Then you can test them later in drug-free conditions, no cocaine, no saline, where the mice have free access to all 3 chambers, and normal mice will tend to prefer to spend more time in the cocaine paired chamber compared to the saline paired chamber. They like cocaine, not surprising. So, and we tested whether chemogenetic inhibition during these context pairings, so inhibition of these LEC projection neurons during these cocaine pairing days, so here and here, during these pink cocaine days. So we tested whether that inhibition during those pairings would affect their preference for a cocaine paired chamber. And so what do we see here? So what we have here is the amount of time that they spend in each chamber. In these mice that express these dread receptors, we're going to call them GI mice. That's the name of the receptor. And these M-cherry controls. So you can think of these as the controls. Now the controls do indeed spend more time in the cocaine paired chamber compared to the saline paired chamber. However, those that have their LEC projection neurons inhibited during that conditioning don't have a preference for that cocaine paired chamber. And then this is just quantified also here on the right. We conducted another similar experiment. However, we inhibited these LEC projections only during that post-test, during the recall of those memories. And what did we find? Well, here we have 3 groups, but you can think of both of these 2 right here as controls. These ones right here express that receptor, but they aren't getting the ligand for the receptor. And these animals here are getting the ligand for the receptor, but they don't express the receptor. It's only this group right here that express both the receptor and the ligand, and thus have the inhibition of these LEC projection neurons. But as you can see, all 3 groups form a preference for that cocaine paired chamber. So these findings suggest that the LEC projection neurons are necessary during the consolidation of cocaine context memories, but are not important during the recall of those memories. So we next sought to determine whether these LEC projections are also important for foot shock context associative memories. So for the task, we tested mice for contextual fear conditioning, which consists of foot shock context pairing, like I described earlier. Mice are then tested a day later for memory recall by measuring their freezing behavior to the shock context. Now, they're not getting shocked, they're just being placed in that same context where they got shocked before. And freezing is a naturalistic fear response in rodents. They'll just like to stop and like kind of like hope no one sees them, and is essentially defined as the lack of skeletal movement. So we first assessed inhibition of these LAC projection neurons during learning on their later memory expression. And so what you're seeing here is the amount of freezing in seconds over an 8-minute test period. And you can see the controls, M-Cherry controls, have a decent amount of freezing, and we see that that's reduced in those that have the LAC projection neurons inhibited. We next did the same thing where we looked at memory recall. What would inhibition of these LAC projection neurons do during recall? Get this to, okay. And so in this case, we inhibited the LAC projection neurons only during that recall test. And what we found is that there was no difference between groups. So these indicate that the LAC projection neurons are necessary for associating salient stimuli like rewards and noxious stimuli like foot shock with context during learning and not during that recall. And so this information that it may be conveying is salient features of the stimuli and context, but do not appear to impart any valence aspects of the stimuli, whether they're positive or negative, just whether they're important. And the LAC to projection neurons also only appear to be important during that consolidation of these memories and not in the recall or expression of a memory. All right, so our story so far. Let's see how much time I have. Okay. All right, we're doing good. All right, so these LAC projection neurons are activated by salient events, rewards, and salient contexts. And we know that they're important for associating salient events with contexts, are important in the consolidation and not the recall of these associative memories. And it may be other hippocampus or other cortices that may be important for that memory retrieval, just not these LAC neurons. So then the question now is, are the LAC and these projection neurons specifically activated by rewards and reward cues? All right, so to test this hypothesis, whether LAC is activated, in this case, by natural rewards, so my technician, Miriam, and undergraduate Richa have been conducting experiments to get at this question, specifically to determine whether LAC neurons are activated by natural reward cues like food. In this case, we're not looking at just the projection neurons, we're looking at the LAC in general. So we conducted this in operant chambers that you can see here, where essentially mice can, they can press a lever to get food, but they're not really actually good at pressing levers, so they just poke their nose into like a little hole, a little recess, while they're in this box. And whenever they poke their nose into these little recesses, they get a little sugar pellet that's delivered into a magazine tray that's in this box here. You can't really see it because it's behind this piece, right? And then whenever they do that, they also get a cue light that's associated with a successful nose poke and reward delivery. And there's actually two nose pokes. There's one that produces the food pellet and the cue, and another that doesn't do anything. So we do this daily, 1-hour sessions, and we do this across 3 different stages. Number 1, we have this self-administration stage where they poke, they get a cue on the active nose poke, they get a cue, and they get a little sugar pellet. We do that for 2 weeks. And then for 2 weeks, they can poke away. They're not getting anything. No cue, no food. And then finally, we do a 1-day test where we simply, anytime they poke, they just get the cue, but they don't get any food. We call that the reinstatement test. So let's look at what that behavior looks like. So this is the number of nose pokes, both active, the ones that produce some consequences, and the inactive that don't do anything. Over all of these sessions, for the self-administration phase, the extinction phase, and that 1-day reinstatement. So what you can see here is that the mice will readily self-administer to get themselves food. These are all somewhat food-deprived mice too, so they're pretty motivated to try to get these sugar pellets. And then when we take away that food delivery, they eventually extinguish and they start nose poking less, which is expected. That first day they get a little frustrated, but then they eventually extinguish their behavior. And then simply by reintroducing the cues themselves, again, no food on this one test, we can reinstate their behavior. And then this is just quantified here on the right for each of those last sessions. So we also took the brains from these mice 90 minutes after the reinstatement session to conduct CFOS histology on slices from the LEC in comparison to mice that were just left in their home cage. And we found that the cue-induced reinstatement of food seeking increased CFOS expression in the tested mice compared to home cage controls. Now, these results could indicate that these LEC neurons are indeed activated during cue-induced reinstatement. However, as we've previously seen, LEC and these LEC projection neurons are activated by contexts. So we weren't entirely sure if the activation was due to the context or the cues or both. So Miriam set out to determine the specific effects of cues in these LEC projection neurons. So to do this, she injected a virus to label the LEC projection neurons, like we talked about before. Then she had them go through the same self-administration and extinction sessions. And then she had 3 groups that all basically differed in the conditions that happened during the actual reinstatement. We had those that got the cue light, just like the previous experiment. Those that didn't get any cues, so like another extinction session. And then those that were just left in their home cage that day. They got everything before then, but they were just left in their home cage that day. And so very interestingly, we found that although only the group that were provided those cues reinstated their responding, that difference between here and here, both the cues and the no cues group had a relatively equal number of CFOS positive LEC neurons. This is in line with our previous findings that salient contexts activate LEC neurons. We're currently still going through the tissue to determine whether we see the same effect in these LEC projection neurons. So TBD on that. More recently, my graduate student Layla has been conducting studies. Sorry, this is my last thing. So my graduate student Layla has been conducting studies to determine whether LEC neurons are specifically activated by cocaine cues. Now, one problem that we see is that we already see this general activation to the context, but we want to see the activation to the specific cues themselves, like that cue light when it comes on. And we already have this background level of activation from the context. So she started conducting fiber photometry, which essentially is this in vivo imaging tool that gives you second-by-second measure of neuronal activation during the behavior, meaning it's way more precise in its resolution compared to CFOS. And we're measuring using this GCAMP, which is this engineered fusion protein. I'm not going to talk about it too much, just we're a little short on time, but I just want to say that this GCAMP is essentially will fluoresce in response to high neuronal activity. And when there's low neuronal activity, this protein won't fluoresce that much. And what does it fluoresce? It fluoresces green, kind of like GFP, but it only fluoresces during high neuronal activity. And it does so very quickly. So it can fluoresce very quickly, like this, but for just short periods of time. Now, we don't get single cell. We only get resolution. We get the total fluorescence from all the cells. I just want to say that. And she measured it while these animals were doing this task. And she essentially used viral vectors to get this GCAMP expression in the LEC neurons. And not just the projections, so just general LEC. And so she conducted the same paradigm as Miriam across the same 3 different stages, and she recorded the activity of the LEC neurons at various time points, including the last day of self-administration, the first day of extinction, last day of extinction, and then that reinstatement test. I'm not going to go over all of those, but I'm just going to show you the results of the reinstatement test, because they're really the most exciting. That's not the only reason. All right, so essentially what you're seeing here is that GCAMP signal. So the higher that is, it means that the more active that neuron is. And we're seeing this across time. And this is very small snippets during the behavior of the reinstatement test. And particularly, it's these snippets around when they poke their nose and they get a cue, but then they don't get any food. Remember, this is the reinstatement test. They only get the cue. And she looks at that specific time window, and then the cue stays on for 5 seconds. So it's basically while the cue is on, immediately after the nose poke. And she's measuring the activity of LEC neurons, just the population signal of all the LEC neurons that express this GCAMP. And what she sees is this increase in response to the cue, indicating, and then this is essentially quantified over here when comparing this 5-second period compared to the 5-second period before it. And we see that there's this increase in activation of these LEC neurons in response to this cue that was previously associated with this food reward. So, LEC neurons are indeed being activated by reward cues. Cocaine? Maybe. We'll find out. We're doing those studies right now. Or Layla's doing those studies right now. Actually, she is doing them right now. So, right now, this minute. All right. And then we are also asking, are these LEC projection neurons necessary for cocaine memories, where we can ask kind of the similar questions that we've been doing using fiber photometry and chemogenetics. So in conclusion, the LEC and these LEC projection neurons are activated by salient contexts and cues associated with rewards and noxious stimuli. That the LEC projection neurons are necessary for associative memory encoding of salient events, and then they may be important for encoding features of salient stimuli, like cocaine and other drugs of abuse. What those features are, we don't know. And then finally, they may be important for drug memories that thus drive craving and relapse. And what I'd ultimately like to do is you see all of these different brain regions here in green that are important in that craving stage. I'd like to add the entorhinal cortex to that once I get a little bit more information. And then we can add that to another brain region that's important in that craving part of that cycle. And if something that we can potentially target that we could use to drive down relapse. So with that, I'd just like to thank all the people that contributed to this. So everyone here has starred, especially Layla, Miriam, and some of the students that were at Michigan State when I was there that helped me really get this off the ground. And I have no conflicts of interest to disclose. Thank you.
All right, fantastic. We'll take questions.
Every time we get these drug-related seminars, I'm horrified. It's really, really fascinating the depth of understanding we're getting on the mechanisms of this reinstatement. But we're presuming there's addictive behavior in the first place, or that there is addiction in the first place because they've used, they've been exposed and used the drug the first time.
Yeah.
What understanding do we have of the mechanisms for not making that bad decision in the first place? And why don't we spend most of our time there? Because we're unraveling a Gordian knot here as opposed to preventing the knot from taking place in the first instance.
That's a great question. I'm going to address it, but then I'm also going to push back. All right, so we do need more prevention. And honestly, things like the massive major indicators of whether someone's going to develop an addiction are like early life stress, childhood history of abuse and neglect, low socioeconomic status, lack of resources. All of those things are major predictive. Now, there are also potential like genetic components as well, which we haven't completely we don't completely understand, or we might happen in some people but not other people. So obviously we should put resources towards prevention. We should always do that. I don't know. I'm a Gen X, so I remember DARE. We had like DARE in my classes. So
I was old when DARE came out.
Yeah, I don't know how well it helped, but I don't know. So, but the problem is we've got people already that have addiction. They need some assistance. If I'm a cocaine addict, I can't go to my doctor and be like, I really need to get off this drug. Can you help me? Someone with diabetes, they can go to their doctor and say, I have diabetes, I don't know how to manage it, can you help me? And the doctor can do that. But doctors can't do anything for someone with cocaine. They're going to say, go to Narcotics Anonymous. That will help you out. Or, talk to a therapist, which that's also helpful, but it's
not curative.
Yeah. And not everyone can do NA. There's people that can, but a lot of people can't. So,
but your trend shows we're doing something wrong because the problem's getting worse and we know more. So we're screwing up somewhere on the front end.
Yeah, and honestly, I would say like just because the opioid overdose crisis we came out with a lot of potential drugs that could target that. Now, most of them are opioid replacements, like other opioids, but they are helping. They're keeping things low, so less people are dying. Hopefully that also translates to less use or maybe more likely to seek treatment, things like that. But we're addressing the immediate need of them dying. And I think it's kind of taken center stage to the other drugs of abuse, like the stimulants, which I think is like, we are readying up for that. We have not a whole generation, but we have many people in like some of the younger generations that are, using these various stimulants so commonly that they'll talk about it with each other. Somebody develops a stimulant addiction, that easily gets translated from a prescription stimulant to like a harder drug like cocaine or methamphetamine, right? Especially if you can't get those prescriptions. We see the same thing with opiates. If they can't get the prescription opiates, they may transition into other harder drugs. So, yeah, I think that's coming.
Thank you.
Sorry. I could go on about this all day, sorry.
Hi, thank you. That's very wonderful talk. I wanted to ask a particular question, but bear in mind about pardon me because if it's just ignore question because I came from a background that is completely never used any direct measurement of neuroactivation. So I was fascinated by the fact that the context that activated the expression is can include both food shock and sugar, like basically food reward. And so food shock, so the contact is not just direct, not just specifically to the, rewarding related contacts, because the food shock can induce a response, if I understand, will induce fear response, I think, from mouse.
Yeah.
So in that case, the context can be both fear-related and also reward-related, assuming that if I understand correctly. So I'm wondering that in that case but the behavior that follow after the context will be different because fear then would lead to avoidance in terms of response from the animals. And so I'm wondering if that's the case, that if there's also entorhinal cortex is involved in the associated responses after. In that case, I don't know if I'm clear, at all.
Let me see if I can. There's no ignorant question, by the way. So I tell them students in my class. Let me see if I can get at what I think you're saying. So first, I want to clarify that we haven't done an experiment where they're getting food and foot shock, like both a reward and a punishment in the same context. These are done in completely different contexts. So we're not making that comparison between those two in terms of the activation at least.
Okay.
But I think, I guess the question is the time after the learning important? Is that what you're asking? Is like after the task is done for like a few hours afterwards during that stabilization period? Is
Yeah.
is that what you were asking?
I'm kinda. I think I'm more like I think I'm more interested in the involvement of like entorhinal cortex and nuclear accumbens in the response after getting the memory consolidated in different contexts.
Okay, yeah. And I would say yes. So that's what essentially like Layla showed in that fiber photometry experiment, right? This is after, they hadn't gotten food for like, I don't know, I'm thinking her experiment, it was like 8 days. Like they were still nose poking, but they hadn't gotten food for like over a week. And just by simply reintroducing the cue, not the food, was enough to activate. So this is way after, like way devoid of the response of working to get the food, right? So they just nose poked and only got a cue light. So so another good question though, and something that we're also trying to get at, is introducing a new type of cue, which is like called the reward availability cue that will happen before the nose poke, that will essentially tell the mice when the reward is available, that is not paired with the actual response itself. We're doing that right now. So we just started introducing that. But that will get at like, is the nose poke itself like causing activation, or is it the response to the cue? And then the reward availability cue will able to tie that differently, because when that comes on, that will basically tell the animal, hey, you can go nose poke now. Whereas before that, if it nose poked, it would result in nothing. So then that will tell us, is this reward availability cue producing activation when the animal's actually not even nose poking at all? So that will kind of get at your question, I think, is what you're asking. Yeah.
Thank you.
Yeah.
Okay, online question coming in. This is a question I think that's related to what you just described. Given that substances of abuse or rewarding substances are different, would the LEC be playing sort of a generalized role with rewards? Let's just restrict it to rewards, or would there be maybe some kind of differences if it was cocaine versus heroin versus alcohol?
That's a great question. And I got to preface this by saying, like, I don't know. It hasn't been tested, right? What do I think? I think, and I've talked to some others about this, like, is it encoding features of like the environment or the context, or is it simply like conveying a signal like, hey, nucleus accumbens, something important happened, but not really like any information about what happened? And I don't know. And then the second question, would it respond similarly to other drugs of abuse? I would suspect that it probably would, right? Just because it's a salient experiment. So if instead of doing the cocaine context, I just do an opioid, I would assume I would see the same type of activation. Or, so I didn't show it here too, we've also just given them cocaine without, like, just taken them out of their home cage, give them a little injection of cocaine, put them back in their home cage. We also see activation just to the drug itself. So not just like drug paired contexts. It's not in here, but yeah. So yeah, I hope that answered that. Oh, it was not your question, the online question. I hope that answered the online question to persons.
Speaking for those online, thank you. We agree. Okay, any other questions before we wrap up? All right, well, we're at time, and it's been a fascinating hour. Thank you so much, Dr.
Thank you all again.
So Andrew will be at UT Dallas and definitely take his courses. We are going to resume in 2 weeks with Dr. Guggen Wig, also from UT Dallas, talking about brain connectivity. So we'll see you then.